You know what is the biggest regret in exam? Not attending the question because you don't know the answer? No, it's not. It's attending the question and end up in writing answer like this. Now look at this answer written by an average student. Completely like an essay type. No important headings, no bullet points, diagram drawn in the corner. Ultimately, this answer ends up in getting 2 or 3 marks. Now look at this answer written by you after following this video. Answer starts with a definition or introduction, all important headings, proper use of tables, flowchart, diagram on the right side, and end up in writing applied physiology. This answer will definitely get you maximum marks in your university exam. In next 15 minutes, I will help you to understand the complete mechanism, draw the important diagram, differentiate multiplier and exchanger, explain the concentrated urine formation, and ultimately present the answer in a proper style format. In university exam, this topic is asked under two ways. Describe the countercurrent multiplier system, what is its role, and discuss the mechanism of formation of concentrated urine and a note on diuresis. First, let me show you the entire answer that we are going to write. First, this is the introduction of the answer, only 3 lines. Then the countercurrent overview, showing two components, multiplier and exchanger. Then multiplication of the single effect, and then countercurrent multiplier. The next slide shows the countercurrent exchanger with the help of Vasa recti, role of ADH in urine concentration, and physiological effect of increase or decrease water intake. And lastly, the applied aspect in the form of table showing water diuresis and osmotic diuresis. If you follow this video till the end, I will help you to remember the entire answer very easily. So let's do it. Concentration and dilution of urine. Let's start with introduction. The main goal of kidney is to maintain volume and osmolality of body fluids within a normal range, and this is done by regulating excretion of two things, water and sodium chloride. So the kidney can produce urine with osmolality as low as 30 milliosmol to as high as 1400 milliosmol per kg. So accordingly, the volume also changes. When osmolality is 30 milliosmol per kg, volume becomes 23.3 liters per day. And when osmolality is 1400 milliosmol per kg, volume becomes 0.5 liters per day. That is concentrated urine. Now to form this concentrated urine, two principal factors are important. First one is antidiuretic hormone and the other one is medullary hyperosmolality gradient. What is medullary gradient? Normal plasma osmolality is about 300 milliosmol per kg, whereas medulla in the kidney is hyperosmolar. That is, its osmolality is higher than the plasma. As you can see from the diagram, this gradient increases progressively from cortex to the medulla, that is from 300 to 1200 to 1400 milliosmol per kg, that is increasing osmolality. And this osmotic pressure is the driving force for reabsorption of water from thin descending segment and collecting duct and ends up in forming concentrated urine. Remember, this is the main principle. Now let's see how this increasing hyperosmolality of medulla is achieved. So it is mainly done by countercurrent system. What is it? It is mainly the inflow which runs parallel to, opposite to, and in close proximity to outflow. That is, it is mainly formed by the U-shaped tubules. You can draw this diagram showing the straight tubule and U-shaped tubule, how U-shape multiplies the heater's effect. For countercurrent system, the renal component are two. First one is multiplier, which occurs in loop of Henle and it creates the gradient, whereas exchanger, it occurs in Vasa recti, it maintains the gradient. Now, countercurrent multiplier. For this, we need to understand the single effect in outer medulla. In this, the thick ascending limb, it actively pumps out sodium chloride. As you can see from the diagram, sodium and chloride comes out actively from thick ascending limb, increasing the osmolality in interstitium, where the same segment is impermeable to water. So water remains inside. That's why this segment is called as a diluting segment. Whereas the descending segment is highly permeable to water. So water comes out very easily. So this part of the segment, the osmolality increases. Now, inner medulla also shows the single effect, but it involves passive sodium transport and urea diffusion. Multiplication of the single effect. As you can see in the beginning, everywhere the osmolality is 300 milliosmol, and then this single effect occurs. In this, the thick ascending limb pumps the sodium chloride outside, making 200 milliosmol gradient inside the tubule, and the interstitium osmolality rises to 400. The water leaves the descending limb to equilibrate with this. And finally, the fresh 300 milliosmol fluid enters, pushing this concentrated fluid down to the hairpin bend. And the cycle repeats. So continuous flow along with active sodium chloride pumping leads to the gradient multiplication. So ultimately, the medullary interstitium gradient increases to 1200 milliosmol. There is a problem with this medullary interstitium. That is, if a straight blood vessel passed through it, it would wash away the sodium and urea gradient. And it is solved by a solution, that is by forming hairpin anatomical arrangement of Vasa recti with a sluggish and very slow blood flow. This entire arrangement is called as counter current exchanger. You can draw this diagram of Vasa recti on the right side. This Vasa recti maintains the hyperosmolarity gradient by two mechanisms. First one, descending limb. In this, the solute diffuses in and the water diffuses out, whereas in ascending limb, the solute moves out and the water diffuses in. And ultimately, it results in blood leaving the Vasa recti at 300 milliosmol, that is, maintaining the gradient. Now let's see the role of ADH in urine concentration. Whenever ADH is high, it causes concentrated or hyperosmotic urine to form, that is, urine with a low volume. And whenever there is low or no ADH, it causes dilute or hypoosmotic urine to form, that is, urine with a high volume. What is the mechanism of action of ADH? Remember, this ADH increases the permeability of collecting duct for water by formation of aquaporin. Now, through these aquaporin, water comes out and forming the concentrated urine. Whenever there is no ADH or low ADH, it makes this tubule impermeable to water. So the water remains inside and it results in dilute urine. This ADH also augments urea cycling into the interstitium. And lastly, ADH also stimulates sodium chloride reabsorption in thick ascending limb. But then, what is the stimulus response effect on ADH, whether it is increase intake or deprivation of water? Let's say it. Whenever decrease water intake or there is deprivation of water, it causes increased plasma osmolality, which stimulate the osmoreceptors in the anterohypothalamus. And response to that, there is increased release of ADH from posterior pituitary, and it increases the water permeability of collecting duct. So there is increased water reabsorption, and it results in decreased urine volume and increased urine osmolality. Whereas, whenever there is increased water intake, it decreases the plasma osmolality. It inhibits the osmoreceptors in anterior hypothalamus. It decreases the ADH secretion from the posterior pituitary and decreased water permeability of collecting duct, results in decreased water reabsorption. So water remain in the urine, increasing the urine volume and decrease urine osmolality. And now here we end up in writing applied physiology in the form of difference between water diuresis and osmotic diuresis. Water diuresis. In this, urine volume is more than 20 liters per day, whereas osmotic diuresis, there also urine volume is more than 20 liters per day because both of them are diuresis. Next, urine osmolality. In water diuresis, urine osmolality is less than 50 milliosmol per kg of water, whereas osmotic diuresis, it is more than 300 milliosmol per kg of water. Specific gravity of urine in water diuresis is always less than 1.010, whereas specific gravity in osmotic diuresis is always more than 1.010. The pathophysiology of water diuresis is absence or reduced ADH secretion, whereas pathophysiology of osmotic diuresis is always unabsorbed solute exceed the tubular maximum, exerting the osmotic effect. Now, water diuresis mainly causes are diabetes insipidus or excessive water drinking, whereas osmotic diuresis, the causes are diabetes mellitus or mannitol administration. Now let's have a quick view to the entire answer of this question of concentration and dilution of urine. Here we have started with the introduction and the two principal factors about antidiuretic hormone and medullary hyperosmolarity gradient. Then we have written about the counter current system overview having two components, counter current multiplier and exchanger. Then in detail about the multiplication of the single effect. Do not forget to draw this diagram having showing the importance of TAL. And the counter current multiplier, the creation of medullary interstitium. TL is the diluting segment. Then we have seen about the counter current exchanger, the role of Vasa recti in maintenance of hyperosmolarity gradient. We also seen the role of ADH in formation of concentrated urine and their mechanism of action. At last, we also seen the stimulus response effect of increase and decrease water intake on this release of ADH. And lastly, we finished with the applied aspect on difference between water diuresis and osmotic diuresis. So you revise this video before exam and then you are done. Now please go and watch these two upcoming videos, first on the counter current mechanism, 5 minutes revision, and second video showing the diagram and flowchart trick to never forget this. Thank you.